Have you ever stopped to think about how your body keeps everything, well, perfectly balanced, even down to the microscopic level? It's this delicate chemistry keeping you going right now.
It really is an intricate dance happening constantly without us even noticing.
Exactly. So today we're doing a deep dive into something fundamental. Acid-base balance. It's one of those silent guardians of your health.
And our goal, really, is to cut through the complex science. We've looked at everything from critical care manuals, newborn physiology studies, even things like liver disease research and lab automation.
Yeah, and even soil science. We want to give you a shortcut to understanding this vital system.
You'll get what pH really means for you, why keeping it stable is so crucial, and what happens when things go wrong.
Get ready for some, hopefully, aha moments about your own inner chemistry.
Let's jump in.
Okay, so let's start at the very beginning. What exactly is pH when we talk about our bodies?
Well, at its core, pH is just a scale measuring how acidic or alkaline something is. Simple as that. In the body, it specifically relates to the concentration of hydrogen ions H++. More H+ means more acidic, less H+ means more alkaline or basic.
Right.
But here's the amazing part. The body operates in this incredibly narrow range, 7.35 to 7.45. That's the sweet spot.
Wow, that's really precise.
It is, and even tiny shifts outside that range can dramatically affect how your cells work, how enzymes function, how ions move across membranes, basically everything.
So deviations have names.
They do. If your blood pH drops below 7.35, it's too acidic. That's called acidemia. If it goes above 7.45, too alkaline, that's alkalemia.
It sounds like our bodies are constantly walking this very fine tightrope. How do they manage to stay so balanced?
They have these incredible internal shock absorbers. We call them buffer systems.
Buffers, okay.
And the absolute star player is the bicarbonate buffer system. It accounts for maybe, 75% of your blood's total buffering power?
75%. So it's a main defense.
Absolutely. It's the body's primary immediate defense against sudden pH swings.
So how does this bicarbonate system actually work? What's the mechanism?
Think of it like a chemical equilibrium. Carbon dioxide, CO2, which we produce all the time, combines with water. This forms a weak acid, carbonic acid.
Right.
Then this carbonic acid can split into a hydrogen ion, H plus A, and a bicarbonate ion, HCO3. The system can shift back and forth to either soak up extra H plus if things get too acidic, or release H plus if it gets too alkaline.
Ah, I see. It's dynamic.
Exactly. And this balance is beautifully described by the Henderson-Hasselbalch equation. For our blood, the crucial takeaway is maintaining a specific ratio.
A ratio.
Yes, for that normal pH of 7.4, you need about 20 parts bicarbonate for every 1 part of that carbonic acid, which is directly related to the CO2 controlled by your lungs.
20 to 1. That's specific.
Very. And who controls these parts? Primarily, the lungs manage the CO2 side, and the kidneys manage the bicarbonate side. They work together.
That's an amazing partnership. Lungs and kidneys keeping the seesaw level.
That's a great way to put it.
But what if the bicarbonate system gets overwhelmed? Are there backup systems?
Oh, absolutely. The body has layers of defense. Beyond bicarbonate, there's the phosphate buffer system. It's particularly important inside our cells and also in the kidneys, helping them excrete acid in urine.
Okay, phosphates too.
And proteins. Proteins throughout your body, especially hemoglobin in your red blood cells, are significant buffers. They can grab onto those extra hydrogen ions.
Hemoglobin. Interesting.
Even your bones play a role, believe it or not. They can release alkaline minerals to help buffer large acid loads over time.
Really? Bones?
Yeah, though it's not ideal long-term as it can affect bone density. But this whole network, bicarbonate, phosphate, proteins, even bone, makes the body incredibly resilient.
Okay, so despite these amazing defenses, sometimes things do go wrong. When these systems get out of balance, we get these acid-base disorders. What are the main types?
Clinicians generally think in terms of 4 cardinal disorders. They stem from either the metabolic side, think kidneys and bicarbonate, or the respiratory side, think lungs and CO2.
And they are.
Metabolic acidosis, metabolic alkalosis, respiratory acidosis, and respiratory alkalosis. Each tells a different story about the imbalance.
Let's break those down, starting with metabolic acidosis. What makes the body too acidic from a metabolic standpoint?
Right, so metabolic acidosis means the primary problem is either a loss of bicarbonate, that HCO3, or a buildup of other acids in the body.
Okay, how does that happen?
Well, you could lose bicarbonate directly, maybe through severe diarrhea, or the body might start producing too much acid. Think of lactic acid buildup during intense exercise or shock, or keto acids in uncontrolled diabetes.
Hmm.
Even things like getting certain IV fluids too quickly, like normal saline, can sometimes cause a specific type called hyperchloremic metabolic acidosis.
So if your body detects this acid buildup, what's its immediate reaction? How does it fight back?
This is where the partnership between systems shines. The body doesn't just sit there. Within minutes, your respiratory system kicks into high gear.
The lungs jump in.
Exactly. You start breathing faster and deeper. There's actually a name for this characteristic breathing pattern, small respiration.
To small respiration.
Yes. The goal is to blow off more CO2. Remember, CO2 acts like an acid in the blood, so getting rid of it helps raise the pH back towards the normal range. It's a rapid compensation.
That makes sense. How do doctors figure out the cause of the acidosis?
A really crucial tool here is calculating the anion gap.
MEN gap.
Yeah, it's a simple calculation using common electrolytes measured in the blood, sodium, chloride, and bicarbonate. It helps distinguish between acidosis caused by gaining an unmeasured acid, like ketones or lactate, which gives you a high anion gap, versus acidosis caused by losing bicarbonate, like in diarrhea, which usually has a normal anion gap.
So it points towards the underlying problem.
Precisely. It's a key diagnostic clue.
Yeah.
And interestingly, even when someone is quite acidic, giving bicarbonate intravenously isn't always the answer. It's usually reserved for very severe cases, like when the pH drops below, say, 7 Anella.
Right. Can you give us a sort of real-world picture? An example.
Sure. Let's talk about lactic acidosis. Lactic acid is a normal byproduct of metabolism, but it builds up when your tissues aren't getting enough oxygen anaerobic metabolism.
Like in shock.
Exactly. Shock, severe heart failure, that's called type A lactic acidosis. But there's also type B, which happens even with normal oxygen levels, maybe related to liver disease, certain cancers, diabetes, or even some medications like metformin.
Okay.
So imagine a patient, maybe a 47-year-old man with type 1 diabetes, who also has severe chronic kidney disease. He might come in feeling unwell, and his blood tests show a pH of, say, 7.23, clearly acidic, along with high blood sugar and high blood pressure. It shows how multiple health problems can converge and really disrupt this balance.
That really illustrates the complexity. Okay, what about the flip side, metabolic alkalosis, when the blood is too alkaline?
Right. Metabolic alkalosis happens when there's a primary increase in that plasma bicarbonate concentration, pushing the pH above 7.45.
And what causes that?
Often, it's due to losing too much acid, specifically hydrogen ions. Think about severe vomiting or having a nasogastric tube suctioning out stomach acid for a long time.
Losing stomach acid.
Exactly. Also, certain diuretics, those water pills like furosemide or thiazides, can cause it. They make you excrete sodium and potassium, but not as much bicarbonate, so the bicarbonate concentration in the blood effectively goes up.
Okay, that covers the metabolic side. Now, let's shift to the lungs. How do they cause problems when they're not working right? Let's start with respiratory acidosis.
So the lungs' main job here is clearing CO2. Respiratory acidosis occurs when they fail to do that effectively. CO2 builds up in the blood, raising the partial pressure of CO2, or PCO2, above the normal range, which is typically 35-45 millimole Ag.
And what causes the lungs to fail like that?
Acute causes could be things that suddenly slow down breathing, like a drug overdose, a head injury, or maybe a severe asthma attack or pneumonia that impairs gas exchange. This leads to a rapid drop in pH and symptoms like anxiety, maybe confusion, drowsiness.
Okay, that's acute. Is there a chronic version?
Yes, chronic respiratory acidosis is common in people with long-standing severe lung diseases like end-stage COPD. Their bodies gradually adapt to chronically high CO2 levels. It's interesting, in these cases, the main trigger telling them to breathe often shifts from high CO2 to low oxygen levels, or hypoxemia.
Fascinating. So if too much CO2 causes acidosis, what about too little? You mentioned hyperventilation earlier.
Right, that leads to respiratory alkalosis. If you hyperventilate, breathe too fast and deep, you blow off too much CO2. This lowers the PCO2, decreases the carbonic acid in your blood, and consequently raises the pH, making it alkaline.
Is that dangerous?
It can be. While some people might not have many symptoms, that low CO2 can actually constrict blood vessels in the brain, reducing blood flow. This might cause lightheadedness, confusion, tingling, or in severe cases, even seizures.
Wow.
It can also mess with electrolytes a bit. Low CO2 can cause potassium levels to dip slightly, and it makes calcium bind more tightly to proteins like albumin. This reduced available calcium can lead to muscle cramps or spasms, that classic tingling feeling.
So it sounds like sometimes these aren't just single, isolated issues, people can have combinations, right? Mixed disorders.
Absolutely. It gets complex. For example, it's actually quite common for critically ill surgical patients to develop mixed metabolic and respiratory alkalosis.
How does that happen?
Well, imagine a patient on a ventilator, which might be set to make them breathe a bit too fast, causing respiratory alkalosis. At the same time, maybe they're losing stomach acid through an NG tube, causing metabolic alkalosis. The two problems add up.
And the body can't compensate properly.
Exactly. In these mixed disorders, the body's usual compensatory mechanisms are often overwhelmed or counteracted, so the pH can drift quite far from normal, making things particularly challenging to manage.
This is a lot to juggle mentally. But I really like that seesaw model analogy you mentioned earlier for the Henderson-Hasselbalch equation. Can you walk us through that again?
Sure, it's a great visual. Think of blood pH as being determined by a seesaw. On one end, you have the metabolic component, represented mainly by bicarbonate, HCO3, controlled by the kidneys.
Okay, kidney and
On the other end, you have the respiratory component, represented by PCO2, controlled by the lungs.
lung end.
For a normal pH of 7.4, that seesaw is perfectly balanced, reflecting that ideal 20-to-1 ratio of bicarbonate to carbonic acid.
Level seesaw, normal pH. Got it.
Now, if one side changes, say, metabolic acidosis develops, meaning bicarbonate drops, that end of the seesaw goes down, the seesaw tips.
Tips towards the CO2 side.
Right. So what does the body do? If the lungs are working properly, they try to level the seesaw by compensating. They blow off more CO2, bringing the respiratory end down to match the lower metabolic end, trying to get the pH back towards normal.
Ah, so the healthy organ tries to correct the tilt caused by the unhealthy one.
Precisely. And the reverse is true, too. If you have respiratory acidosis, high CO2, the kidneys will eventually compensate by retaining more bicarbonate to try and level things out.
That makes it much clearer. How quickly do these compensations happen?
It varies. Respiratory compensation for a metabolic problem starts within minutes, as we discussed with small breathing, and it's usually well underway within 12 to 24 hours. Metabolic compensation by the kidneys for a respiratory problem is much slower. It can take several days to fully kick in.
Right. Lungs fast, kidneys slow. That seesaw model really helps visualize the interactions. How do doctors actually track all this in a patient?
The cornerstone is a test called an arterial blood gas analysis, or ABG. It's a very common test, especially in critical care.
ABG. What does it measure?
It directly measures the pH, the partial pressure of carbon dioxide, PCO2, and the partial pressure of oxygen, PO2, in arterial blood. From these, and usually other electrolytes, the lab can calculate the bicarbonate level, HCO3, and also something called the base excess, or deficit, which gives another view of the metabolic component.
So give you all the key players on the seesaw.
Exactly. Interpreting an ABG is a structured process. First, look at the pH. Is it acidemia, 7.35, or alkalemia, 7.45?
Step 1. Acid or alkali?
Step 2. What's the primary driver? Look at the PCO2. If it's abnormal in the direction opposite to the pH change, for example, high PCO2 and acidosis, it's likely a primary respiratory problem. If the PCO2 is normal or changing in the same direction as the pH, look at the bicarbonate base excess. If that's abnormal in the same direction as the pH change, for example, low bicarbonate acidosis, it's likely primarily metabolic.
Okay, find the main culprit, lungs or metabolism.
Step 3. Is there compensation? Look at the component that isn't the primary driver. Is it changing in the expected direction to try and correct the pH? For example, in metabolic acidosis, low bicarb, are the lungs compensating by lowering the PCO2? Or in respiratory acidosis, high PCO2, are the kidneys compensating by raising the bicarb?
Check if the other side of the seesaw is trying to help.
Precisely. The systematic approach helps clinicians quickly assess the patient's status, determine the type of disorder, see if compensation is adequate, or identify if it's actually a mixed disorder where multiple things are going wrong at once.
That makes sense. Are there certain groups of people who are particularly vulnerable to these imbalances, where maybe their buffer systems or compensatory mechanisms aren't as robust?
Absolutely. Newborn infants, especially premature babies, are a key example. They are quite prone to metabolic acidosis.
Why is that?
Well, their kidneys are still developing. The glomerular filtration rate, how quickly the kidneys filter blood, is lower, and the kidney tubules, which are responsible for reabsorbing bicarbonate and excreting acid, aren't fully mature. They just don't have the same capacity as an adult kidney to handle an acid load or conserve bicarbonate.
So their kidneys are still learning the ropes.
In a way, yes. Interestingly, a recent study, actually the first of its kind, looked at blood gas values in healthy full-term newborns right after birth using capillary blood. They found the average pH was around 7.30 and PCO2 was quite high, about 52 mmHg.
That sounds acidic compared to the adult range.
It is, slightly. But by 48 hours later, those values had shifted closer to the adult normal range, pH around 7.39, PCO2 around 38.7 mmHg. It shows just how rapidly their physiology is adapting after birth. Having these normal reference values for newborns is incredibly important, especially if cord blood wasn't available at delivery to help doctors spot potential problems early on.
That's fascinating. We usually focus on lungs and kidneys, but you mentioned the liver earlier. How does liver disease, like cirrhosis, impact acid-base balance? It seems less direct.
It's surprisingly complex. Cirrhosis throws several wrenches into the works. One common finding, seen in nearly 40% of cases, is actually respiratory alkalosis. The patient is hyperventilating.
Hyperventilating. Why?
The exact reasons aren't fully clear, but it might be linked to low oxygen levels, hypoxemia, which can stimulate breathing, or possibly high ammonia levels, hyperammonemia, which is common in liver failure and can also affect the respiratory drive.
Okay, so the liver dysfunction triggers the lungs to overcompensate.
It seems so. And you also see mixed disorders. About 30% of cirrhosis patients have both respiratory and metabolic alkalosis. The metabolic part can be driven by several factors. The liver isn't making as much urea, which affects acid handling, hormonal changes like secondary hyperaldosteronism, and very commonly the use of loop diuretics to manage fluid buildup, which, as we said, can cause bicarbonate retention.
Wow, so many interconnected factors.
Even low albumin levels, common in cirrhosis because the liver makes albumin, can play a role. Albumin is a weak acid, so having less of it might predispose someone to alkalosis. It's a real tangle.
It really underscores how central the liver is, even to this balance. Thinking about how the body handles an acid load, it's not just one reaction, is it? You mentioned a multi-step process.
That's right. Research, including some classic studies done in dogs where they were carefully infused with acid, revealed this really elegant three-phase response.
3 phases, okay. What's phase 1?
Phase 1 is immediate buffering, mostly in the extracellular fluid, the blood plasma, and the fluid surrounding cells. Bicarbonate is the main player here, neutralizing a bit over half, maybe 56% of the initial acid load almost instantly.
Immediate shock absorption by bicarbonate. Phase 2.
Phase 2 involves the buffers inside the cells taking over. This happens more gradually over hours. Intracellular buffers, including proteins and phosphates within cells, start neutralizing the acid that has moved into the cells. Over about 24 hours, these intracellular buffers handle a significant chunk, maybe increasing their share to about 3/4 of the total acid load neutralized up to that point. This often involves alkali, like bicarbonate, moving out of cells to help buffer the blood too.
So first outside the cells, then inside. What's phase 3?
Phase 3 is the long game, played by the kidneys. They're responsible for the ultimate excretion of the excess acid from the body. This is a slower process, taking anywhere from 2 to maybe 8 days for a moderate acid load.
Days.
Yes. The kidneys do this primarily by excreting hydrogen ions bound to ammonia, forming ammonium chloride, and other substances called titratable acids in the urine. This phased response, immediate buffering, then intracellular buffering, then final renal excretion ensures that the acid challenge is completely dealt with and the body's stores of buffer are eventually replenished.
That's incredibly sophisticated. A multi-stage defense. Thinking about inputs, can something as basic as nutrition affect this balance?
Oh, definitely. Especially in critically ill patients. For instance, providing too many calories, particularly carbohydrates, can actually increase the amount of carbon dioxide the body produces through metabolism.
More CO2, which could lead to acidosis.
It could potentially worsen or lead to respiratory acidosis, making it harder to wean someone off a ventilator.
Interesting. What about malnutrition?
Malnutrition is a huge issue. Some estimates say 1 in 3 hospitalized patients in the U.S. is malnourished, leading to longer stays and more complications. In these patients, acid-base imbalances can sometimes be nudged in the right direction using specialized intravenous nutrition called parenteral nutrition.
How so?
Well, the formulation can be adjusted. For example, if a patient has metabolic acidosis, clinicians might increase the amount of acetate salts in the IV nutrition. Acetate gets metabolized by the body into bicarbonate, helping to counteract the acidosis.
Providing a bicarbonate precursor.
Exactly. And conversely, if a patient has metabolic alkalosis, they might increase the chloride salts in the mix. But it's important to remember, nutrition alone isn't going to fix severe acidosis. It's just one part of the overall picture.
Right, a tool in the toolbox. Let's shift gears a bit. beyond the body, how are scientists applying these principles of pH control, maybe using technology?
Well, precise pH control is critical in tons of settings. Think cell cultures, biotech manufacturing, chemical reactions. Doing it manually can be tricky and slow.
Yeah, I can imagine.
So there's some cool new tech. One example is a silicon-based impedance chip. It offers a really fast, reagent-free way to monitor the concentration of buffers after they've been diluted.
How does that work?
It measures electrical impedance, and researchers found a specific parameter called ρC that changes linearly on a logarithmic scale as the buffer concentration changes.
Hmm.
And the neat part is this relationship seems to hold true regardless of the specific pH or the volume of the sample.
So it directly measures buffer strength, not just pH.
Essentially, yes, it gives you a handle on the buffering capacity. This kind of tech could be huge for automating liquid handling systems in labs or industry, providing real-time feedback to ensure buffer concentrations are exactly right.
That sounds like a major step up for consistency in sensitive experiments.
It really is. And alongside monitoring, there's always ongoing research into developing better buffers. For example, one study looked at buffers used for in vitro digestion studies, simulating digestion in a lab. They found that a specific phosphate bicarbonate buffer, adjusted with citric acid, was much more stable at maintaining the target pH over time compared to using phosphoric acid. It had less pH drift and didn't seem to interfere with the digestion process itself.
So constant refinement of the tools.
Exactly. Always looking for more stable, more effective ways to control pH.
What about really complex situations like mixtures with multiple buffering agents? Predicting pH changes, there must be a nightmare.
It absolutely can be. Traditional chemical models can really struggle with those complex, multi-buffered systems where you have several different substances all contributing to the buffering.
So what's the solution?
This is where machine learning is making inroads. Researchers are now using robotic platforms combined with active machine learning algorithms.
Robots and AI for pH.
Kind of, yeah. They use algorithms, often Gaussian processes, to learn the titration curve, how the pH changes as you add acid or base for these complex mixtures in real time. The system can then predict how much acid or base is needed to reach a specific target pH much more efficiently than just trial and error.
That sounds powerful.
It is. They've shown it can work well even in mixtures with up to 4 different buffers, like citrate, phosphate, ammonium, and acetate all mixed together. And using techniques like transfer learning, the system gets smarter over time. It learns from previous experiments, making adjustments faster and more accurate, even if you don't know the exact concentration of everything in the mix.
Wow, that could have huge implications for chemical manufacturing, maybe even drug development or personalized medicine down the line.
Potentially, yes. It's a really data-driven approach to a classic chemistry problem.
Okay, one last area, bringing it back to the natural world. How does this concept of buffering play out in something as fundamental as soil?
Soil pH buffering capacity, often called pHBC, is absolutely critical for healthy ecosystems. It determines how resistant the soil is to changes in acidity, perhaps from acid rain or certain fertilizers.
So soils have their own buffer systems.
They do, but the mechanisms differ. In soils that contain carbonates, think soils derived from limestone or chalk, the buffering is mainly chemical. The carbonates simply react with and neutralize added acid, keeping the pH relatively stable until the carbonates are used up.
Okay, like a built-in antacid.
Pretty much, but in soils without carbonates, which is common in many areas, the buffering capacity is different. It's primarily determined by something called the cation exchange capacity, or CEC.
Cation exchange capacity. What's
Yeah.
that?
It's basically the soil's ability to hold on to positively charged ions, like calcium, magnesium, potassium, but also hydrogen ions, H+. Soils with a higher CEC can absorb more H+ ions from the soil water, resisting a drop in pH more effectively. It's more of a physical-chemical process involving the surfaces of clay particles and organic matter.
So different soils buffer acid in fundamentally different ways.
Exactly. Carbonate soils rely on chemical neutralization, while non-carbonate soils rely more on their cation exchange properties. Understanding which mechanism dominates in a particular soil is key to managing soil health, predicting how fertilizers will behave, and understanding how ecosystems might respond to environmental changes like acid deposition. It directly impacts plant growth and overall ecosystem resilience.
That's incredible. It really shows how universal these principles of buffering and balance are.
From our own cells to the soil beneath our feet.
So we've covered a lot of ground today.
Wow.
From those fundamental buffers like bicarbonate working tirelessly in our blood to that sophisticated three-step defense the body mounts against acid.
Yeah, and we saw how critical this balance is for vulnerable newborns, the complex ways it gets disrupted in liver disease.
And even how cutting-edge tech like robotics and machine learning are being used to manage pH with incredible precision, right down to understanding the buffering in soil.
It's a system that's often invisible, working silently in the background.
But hopefully, you now have a much deeper appreciation for just how powerful and essential this acid-base balance truly is for governing life.
Absolutely.
So here's a thought to leave you with. The next time you hear someone mention pH, maybe think about what other silent guardians might be at work, both inside you and in the world around you. These systems maintain delicate balances we barely notice until they're disturbed.
And perhaps could a deeper understanding of these subtle balancing systems unlock new solutions, maybe for personalized health or managing our environment more sustainably?
Lots to think about.
Okay.
Keep exploring, keep questioning. This deep dive is really just scratching the surface of life's intricate chemistry.
Until next time, stay curious.